Usually when we talk about a medical diagnosis, there's this expectation of absolute crystalline precision.
Right. It feels almost like engineering.
Exactly. You break your arm, the x-ray shows that jagged white line on the screen, and the doctor just points at it and says, there it is.
Yeah, the bone is broken, and here's how we fix it. It's very static.
And, honestly, it's comforting.
Oh, absolutely. As humans, we like our problems to be highly visible. We want them easily categorized into broken or not broken.
We really want the body to act like a simple machine where you just, swap out a bad part.
But then you step into the world of tissue regeneration, and suddenly that static X-ray machine is completely useless.
Right, because we're no longer looking at a simple machine. We're looking at a biological landscape that is fiercely dynamic.
That's constantly assessing itself.
Yeah, and capable of seemingly impossible feats of reconstruction. So today we are focusing on the ultimate biological superpower right inside your own body, the liver. It is truly the undisputed champion of human organ regeneration.
It really is. You can surgically remove up to 70% of a healthy liver, and within weeks, it will completely rebuild itself back to its original size and function.
Which is just wild to think about.
It's a level of cellular plasticity that we usually only associate with, starfish or salamanders.
And the ancient Greeks actually had an intuition about this thousands of years ago.
Oh, the Prometheus myth.
Yes. Think of the myth of Prometheus. He stole fire from the gods to give to humanity, and Zeus's punishment was just brutal.
Chained to a rock.
Right, chained to a rock. And every day an eagle would fly down and eat his liver. And every night the liver would magically regrow,
setting him up for the exact same torture the next day.
It's such a gruesome piece of mythology, but scientifically, it is remarkably accurate regarding the liver's raw capacity.
The Greeks somehow grasped that the organ could regenerate, which is mind-blowing.
But modern science has spent decades trying to figure out the deeper mystery here. We are just now figuring out exactly how the liver knows when to start regrowing, and crucially, how it knows when to stop.
Right. Biologists refer to this as the hepatostat.
The hepatostat.
Yeah, it's this invisible internal size control mechanism. Think of it like a thermostat in your house, but instead of regulating temperature, it regulates total liver mass.
And that brings us to the source material for this deep dive. We have our hands on a groundbreaking 2026 research article published in the journal Science by Zhang and a large team of colleagues.
It's a fascinating paper.
It really is. So our mission today is to show you exactly how the liver orchestrates its own regrowth. And spoiler alert, it's not just relying on chemical signals floating around in your blood.
No, it is using literal mechanical physics.
Okay, let's unpack this.
So to really grasp the mechanics of the hepatostat, we have to start with the architecture of the organ itself.
Right. The liver is not just a uniform, homogeneous block of meat.
Exactly. It is highly organized into a repeating hexagonal structure called a lobule.
A lobule, okay.
And we divide this lobule into what we call a zonal map. There are 3 distinct zones, defined entirely by the direction of blood flow and their specialized metabolic functions.
Okay, to help you visualize the zonal map at home, I want you to picture a sprawling, carefully planned city. So Zone 1 is your industrial energy sector.
The periportal zone.
Right. The blood enters the liver here first, bringing oxygen and nutrients straight from the digestive tract.
And because these cells get the most oxygen, they're the power plants. They handle heavy energy production and metabolism.
But that heavy industry comes with a cost, right?
A huge cost. Because Zone 1 is churning through so much oxygen to produce energy, it generates a massive amount of oxidative stress.
Which is essentially biological exhaust or smoke from those power plants.
Exactly. Then, on the complete opposite side of the city, where the blood is exiting the liver, you have Zone 3.
The pericentral zone.
Right.
And Zone 3 is your waste management and heavy detox center.
Yeah, by the time the blood gets here, the oxygen is mostly depleted, but the toxins remain.
So these cells are packed with specialized enzymes to process drugs, alcohol, and toxic metabolic byproducts.
They are dealing with the heaviest, most dangerous garbage the body produces.
Which leaves us with Zone 2, the mid-lobular region.
Yeah, it sits right in the middle, perfectly sandwiched between the high oxygen energy sector of Zone 1 and the toxic waste processing plant of Zone 3.
So Zone 2 is basically the quiet residential suburb. Not too much heavy industry, not too much toxic waste.
Exactly. Now, the researchers in this science paper wanted to know which of these 3 zones is responsible for rebuilding the city when disaster strikes.
And they used a staggering piece of technology to figure this out called single-cell transcriptomics.
Right, which is revolutionary. Instead of grinding up a piece of tissue and getting like an average reading of what the cells are doing.
Like a biological smoothie.
Yeah, exactly. Instead of that, this technology isolates every single cell and reads its exact RNA instruction manual in real time.
Which gives incredible detail.
It does. And they paired that transcriptomic data with something called lineage tracing in mouse models.
Okay, you can think of lineage tracing as a biological GPS tracker, right?
Exactly. The researchers genetically engineered mice, specifically using the Krelok system, to permanently attach a fluorescent tag to cells that express a specific surface protein called DPP4.
And what's brilliant about this technique is that when those tagged cells divide, their daughter cells also carry the glowing tag.
Yes, you can track the entire family tree of a cell over time.
And the results were just stunning. When they tracked those glowing markers, they found that these DPP4 positive cells localized almost entirely in zone 2.
Our quiet suburb.
Right. They are the secret regenerative engine of the entire organ. When the liver is injured, it is these specific zone 2 cells that wake up, rapidly multiply, and physically spread outward to rebuild both the industrial sector of zone 1 and the waste management sector of zone 3.
It's an elegant system.
But wait, I have to push back on this logic for a second.
Okay, go ahead.
Why zone 2? I mean, if the blood enters at zone 1, bringing all the systemic nutrients and potential injury signals from the rest of the body, wouldn't those zone 1 cells be the first to get the "we need to grow" memos?
You would think so.
Yeah. Why rely on the middlemen hiding out in the suburbs to do the heavy lifting of regeneration?
It comes down to cellular preservation and really evolution.
Okay.
Zone 1 is constantly battling that massive oxidative stress we mentioned from energy production.
The power plant exhaust.
Right. And Zone 3 is taking a relentless beating from toxins, making it highly susceptible to necrosis or cell death.
Ah, I see.
So if a biological system is going to maintain a reserve of pristine stem-like cells ready to deploy at a moment's notice to rebuild an entire organ, it cannot place those cells in the active war zones.
That makes perfect evolutionary sense. Zone 2 is the perfect anatomical safe haven?
Exactly. It is geographically shielded from both the oxidative fires of Zone 1 and the toxic wasteland of Zone 3, keeping the DPP4 positive cells undamaged and primed to divide when the alarm sounds.
Wow. Okay, let's do a quick recap for you listening at home so we can lock this in before we zoom in closer.
Good idea.
First, the liver has distinct zones based on blood flow, like a city. Second, Zone 2 is our quiet suburb. It acts as the safe haven backup generator for the organ. And third, DPP4 is the specific protein marker that identifies these elite regenerative cells. Keep that map in your mind.
Because this transition brings us to the core mystery of the paper.
Right, we know where the backup generators are, Zone 2. But how do these specific cells in the quiet suburb even know that the liver has been injured?
We need a causal trigger. To find it, the research team studied what happens immediately after a 70% partial hepatectomy.
That is the surgical removal of 70% of the liver mass, right?
Yes. It is a massive, sudden physiological insult. The remaining 30% of the liver suddenly has to process 100% of the body's blood flow and metabolic demands.
And before any cells even have time to divide or multiply to replace the missing tissue, the remaining liver cells, the hepatocytes, undergo an incredible physical transformation.
They instantly swell up.
Yeah, they gorge on fluid and nutrients, physically expanding their cellular volume to compensate for the sudden loss of mass. This process is called cellular hypertrophy.
The biomechanics of this swelling are extraordinary. The researchers deployed highly sophisticated tools to measure exactly what this swelling feels like at the microscopic level.
How do you even measure that?
They used atomic force microscopy, or AFM.
Okay.
AFM doesn't just look at cells. It literally touches them. It uses an infinitesimally small cantilever.
Which is like a microscopic diving board with a laser bouncing off the back of it, right?
That's a great way to picture it. So this tiny diving board gently presses down on individual cells in the liver tissue. By measuring exactly how much the cantilever bends, they can calculate the precise stiffness or elastic modulus of the tissue.
The numbers from the AFM are wild. They found that after the surgery, the tissue stiffness, specifically in our Zone 2 suburb, nearly doubled.
It went from a baseline of 2.43 kilopascals to a highly rigid 4.53 kilopascals.
And to visualize that molecular stiffness in real time, they also utilized a specialized fluorescent probe called Flipper-TR.
Right, Flipper-TR. This is a molecule that wedges itself into the lipid membrane of the cell.
And when the membrane is relaxed, it emits one specific wavelength of light.
But when the cell swells and the membrane lipids are pulled tightly apart, the probe physically flattens out, completely changing its fluorescent signal.
So using this probe, the researchers watched a massive spike in membrane tension light up almost exclusively in zone 2.
Yes.
It's like overinflating a water balloon. You know when you attach a balloon to a faucet and you put just a little too much water in?
Oh yeah, it feels like it's going to pop.
Exactly. The rubber goes from being soft and pliable to incredibly tight, shiny, and stiff. That is exactly what is happening to your remaining liver cells.
They are physically stretching to their absolute limits, turning zone 2 into a localized biomechanical hotspot.
Let's pause to recap this critical step. Injury leads to sudden cell swelling, which creates immense physical tension, specifically in zone 2. Have you ever thought about your cells literally feeling stretched? Like, how does a microscopic biological entity actually sense that physical tightness and translate it into an instruction to start dividing?
Well, what's fascinating here is the absolute star molecule of this entire process.
Yes, piezo1.
Piezo. It is a mechanosensitive ion channel. Instead of waiting for a chemical key to unlock it, piezo1 responds exclusively to physical force.
Structurally, it looks almost like a 3-bladed propeller embedded directly into the outer lipid membrane of the cell.
Right, and when the cell membrane stretches, the physical pulling force yanks on those propeller blades, flattening the structure and ripping open a pore in the center of the channel.
And the liver's architectural brilliance is on full display here. The researchers mapped the location of these piezo1 channels and found that they aren't scattered randomly.
No, they are highly enriched, specifically in those DPP4 positive cells in zone 2.
The liver perfectly aligns its highest mechanical tension, that water balloon stretching, with its highest density of physical tension sensors.
It's an impeccably localized alarm system.
It really is.
And to prove beyond a doubt that piezo1 was the specific switch, the researchers employed chemical tools to manipulate the channel directly.
They used an activator named YODA1.
Right, YODA1 acts like a wedge, physically forcing the piezo1 propeller open, even without any membrane stretching.
And conversely, they used a blocker called DUCA1, which essentially glues the channel shut, preventing it from opening no matter how much tension the cell experiences.
I have to say, the Star Wars references in the nomenclature are fantastic.
They are. But the mechanism is even better. I like to visualize piezo1 like a booby-trapped door in an Indiana Jones temple.
Oh, I like that.
The cell membrane is the stone floor. When it stretches, it's like stepping on a hidden pressure plate. The mechanical force triggers the trap, the door flies open, and a flood rushes into the room.
Only in the cell, that flood isn't water or sand. It's a massive influx of calcium ions.
Right, so a quick recap for you. Physical stretch equals the opening of the piezo1 door, which equals a massive flood of calcium into the cell.
And in cellular biology, a sudden concentrated flood of calcium is the universal klaxon alarm.
It tells the cell's internal machinery that a major event is occurring and immediate action is required.
The moment that piezo1 door is pulled open by the tension, the calcium gradient forces ions rushing into the intracellular space.
Okay, the door is open, the calcium is inside. But what does the calcium actually do to construct a new liver? It has to talk to the cell's DNA somehow.
Right, the calcium initiates a beautifully complex downstream domino effect. It binds to and activates a specific protein called NFATC3.
NFATC3.
Yes. NFATC3 is a transcription factor. You can think of a transcription factor as a specialized messenger carrying a VIP all-access pass.
Okay, I'm following.
In its resting state, NFATC3 just hangs out in the main body of the cell. But when calcium binds to it, the protein actually changes its three-dimensional shape.
And this shape change exposes a specific sequence of amino acids called a nuclear localization signal, which is the VIP pass.
Exactly. With that pass exposed, NFTC3 travels straight into the cell's nucleus, which is the highly protected vault where all the DNA instructions are stored.
And it scans the genome and searches for one very specific gene, right?
Yes, IGFBP2, which stands for insulin-like growth factor binding protein 2.
So NFATC3 binds directly to the DNA and turns the IGFBP2 gene on, initiating the production of this vital protein.
And IGF-BP2 is the ultimate green light.
It's the specific effector protein that triggers the cell cycle, officially commanding this stretched-out, tense hepatocyte in zone 2 to finally divide and multiply.
That is the complete molecular relay.
But wait, I need to pause and look at this from a systems engineering perspective for a second.
Sure.
Because this seems like an incredibly elaborate Rube Goldberg machine. I mean, the cell swells, which stretches the membrane, which pulls open a physical door, which lets in calcium, which changes the shape of a messenger, which enters the nucleus to turn on a gene to finally make the cell divide.
It's a lot of steps.
Why go through all this mechanical trouble? Couldn't the cell just use standard chemical growth factors floating in the blood to trigger IGF-BP2 directly?
Well, if we connect this to the bigger picture, the necessity of this mechanical pathway becomes obvious. It serves as a vital, non-negotiable physical checkpoint.
Ah, the hepatostat in action.
Exactly. It ensures that the liver only initiates massive cellular division when there is actual physical room and a mechanical need for it to grow.
Because if the liver grew based purely on circulating chemical signals, it might become deaf to its own spatial boundaries.
Right. It would just keep responding to the chemicals in the blood, expanding continuously without any breaks.
And continuous, unbounded cellular division is the literal definition of a tumor.
Precisely. By inextricably tying the growth trigger to the physical stretching of the remaining tissue, the liver ensures it only multiplies when there is missing mass.
So the moment the liver regrows to its normal volume, the cellular crowding stops the cells from stretching.
The tension drops.
The piezo-1 channel snaps shut, the calcium stops flowing, and the growth halts entirely.
It is a flawless, self-regulating biomechanical loop.
That is incredible. Okay, let's recap this full molecular cascade so we don't lose the thread. Tension opens the piezo-1 channel, calcium rushes in. That activates the NFATC3 messenger, which goes into the nucleus to turn on the IGF-BP2 gene, which finally tells the cell to divide.
Perfectly summarized.
Right.
Now, in the scientific method, mapping out a theoretical machine like this is an incredible achievement.
But to truly prove the machine works exactly the way you hypothesize, you have to manipulate it.
Yes, you have to break the machine to see if it stops working, and you have to supercharge it to see if it runs faster.
And the researchers did exactly that. First, they broke it.
They genetically engineered DPP4 piezo-1 KO mice.
KO meaning knockout.
Right. In these mice, they deleted the piezo-1 gene, but they only deleted it in those specific DPP4 positive zone 2 cells. The rest of the liver was completely normal.
And the physiological results were stark and immediate. Without the piezo-1 mechanosensor, there was total regenerative failure.
After surgical removal of liver tissue, the zone 2 cells swelled up just like before, but they were effectively blind to the tension.
They stayed as isolated single units, utterly failing to form the multicellular clusters necessary for architectural regrowth. The overall liver mass simply couldn't recover.
They even took it a step further and tested this by inducing a toxic injury instead of a surgical one.
Right, they injected the mice with a chemical called CCl4.
Which is a potent toxin that specifically targets and destroys Zone 3, the waste management sector.
And in a normal liver, the zone 2 cells sense the loss of their neighbors, wake up, and expand to replace the dead zone 3 tissue.
But in the mice without piezo-1, the zone 2 cells did nothing.
Because the physical tension sensor was gone, they couldn't detect the structural collapse happening right next door.
Exactly. The liver suffered from persistent, unchecked necrosis, widespread, uncontrolled cell death, that it was completely powerless to heal.
But here's where it gets really interesting. Because breaking the system wasn't enough for this team.
No, they wanted to see if they could master it.
They introduced a specific gain-of-function mutation into the mice known as the R2482H mutation.
What this mutation does is subtly alter the structural kinetics of the piezo-1 channel.
How so?
Well, when the membrane stretches and the propeller blades pull the pore open, this specific mutation prevents the channel from closing normally.
Oh, wow.
Yeah, it forces the pore to stay open significantly longer than it ever would in a wild-type cell.
It's like taking that booby-trap door from Indiana Jones and kicking a wedge under it. The door gets stuck wide open, allowing a massive, prolonged, and continuous flood of calcium to pour into the cell.
And the biological outcome of this prolonged mechanical signaling was explosive. The researchers observed incredibly fast, hyper-accelerated liver regeneration.
These cells multiplied at an unprecedented rate, restoring the liver mass and healing the damaged tissue vastly quicker than in the control subjects.
The system didn't just work, it was highly tunable.
Okay, final recap of the proof here. No piezo-1 means absolutely no healing, But hyperactive piezo-1 means super healing.
It's a dramatic confirmation of the pathway.
So, bringing this all together, what does this all mean for you listening right now? Why dedicate a deep dive to the squishiness of mouse livers and the shape of microscopic protein channels?
Because this research shatters a long-held biological assumption.
Exactly. It proves that the physical forces acting on your body, the tension, the stretching, the cellular stiffness, are just as critical to your health, healing, and survival as your genetics or your blood chemistry.
This raises an important question regarding the entire future of hepatology and internal medicine.
Right. Currently, our approach to treating severe liver diseases, conditions like cirrhosis, severe hepatitis, or fatty liver disease, is almost entirely chemically focused.
We prescribe drugs to reduce chemical inflammation, or we try to block specific chemical receptors in the blood.
But this paper completely changes the paradigm. It reveals that if we want to help a failing liver regenerate, we can't just look at the chemical exhaust. We have to look at the structural foundation.
We might need to target this exact PO11 IGFBP2 mechanical axis. We need to focus on the physics of the organ.
If pharmacologists can design highly targeted therapies that safely modulate this mechanosensor in humans,
Perhaps a localized version of that YOTA1 activator.
yeah, we could potentially wake up the liver's natural regenerative reserve on demand. We could kickstart the hepatostat before the organ reaches complete failure, offering a lifeline to patients waiting on transplant lists.
It's an incredibly hopeful frontier.
It really is. And that leads me to a final, slightly mind-bending concept for you to mull over.
Oh, here we go.
If piezo-1 acts as the liver's physical size sensor and internal stretch detector, could we one day move beyond chemical drugs entirely?
That is a fascinating thought.
Right. Could we use targeted, non-invasive physical therapies to heal our internal organs? Imagine a future where a doctor uses highly focused, localized sound waves or specialized internal acoustic vibrations to physically manipulate the stiffness of a diseased liver from the outside.
Using frequency to target mechanosensors.
Yes. Could we use vibration to literally jiggle and stretch these zone 2 cells, physically pulling the piezo-1 doors open and tricking the organ into aggressively regrowing itself without ever prescribing a single pill?
It completely blurs the line between biology and mechanical engineering.
It really does. So next time you stretch your muscles, remember that on a microscopic level, your cells are feeling that physical tension too, and it might just be the very mechanism keeping you alive. Thank you for joining us on this deep dive into the architecture of your own biology. Keep questioning the incredible mechanics hiding inside you, and we'll see you next time.